I don't know why. It's a modern MacBook. Should work, but it doesn't. Yeah, is it the PDF file?
No, it's the actual PowerPoint file.
Yeah, seems working.
It looks like it's working.
Yeah.
Put it in viewer and see what it looks like.
It seems blue rather than white.
Look at that.
Yeah. Okay, so can I have the first one? How can I switch? Where's the pointer?
Pointer is that green button right there.
The green one. This
Yeah.
to move forward.
This way. So,
Okay.
and then that should be move forward. So there you go.
Okay.
And then the green is the.
Okay. Okay, finally. Okay.
I think it should be already on. Let me see.
No, it doesn't work.
See here. Oh, you're right. He hasn't turned it on because it's on there. Go ahead and give it a test.
Yeah. I will try to stay in time. Good morning. Sorry for the technical problems. I wanted to use my MacBook, but was not compatible. Good morning, everyone. And so today I'm going to talk about the sources of DNA found on the Shroud of Turin. First of all, let me thank the organizers, especially Bob Racker for the kind invitation and for the opportunity to attend for the first time a Shroud conference like this one. Secondly, let me tell you that I'm not a syndenologist. I'm a geneticist working at the University of Padua, which is an historical town very close to Venice, Italy. It is one of the oldest universities in Europe. It was established in 1222, so almost 8 centuries ago. And it is the university where Galileo Galilei was teaching for many years. So today we'll present the main findings from DNA analysis we performed on genomic DNA samples isolated from microscopic particles and fibrils vacuumed from the backside of the shroud. We published our article almost 2 years ago, and it is, let me tell you that it is in the top 100 read articles in the last 2 years. Some background. I know that everyone here in the audience knows the long journey of the Shroud much better than I do, but this information is very important for understanding our results. So, according to many scholars, you know that the long journey of the Shroud began in Jerusalem almost 2,000 years ago. After concealment for several years, it was moved to Edessa, now Sanliurfa, in the modern country of Turkey, and then to Constantinople in 944. Later, the Shroud was owned by the Byzantine emperors, but disappeared during the sack of Constantinople. So we are here in 1204. After this event, information on the Shroud is very scanty. We know that it reappeared in France at Liry around the year 1356. Then it was moved to Chambéry from 1453 to 1578. And subsequently, it passed into the hands of the Dukes of Savoy. The Shroud was likely hidden in southern Italy during the war periods, and then it was brought to Turin in 1578 and later placed in the city cathedral, where it has been permanently conserved from 1694 to the present. So here, the long journey of the shroud. From a temporal and geographical point of view, we could split the long journey of the shroud into a middle or Near East age and area before the sack of Constantinople, 1204, and into a Western European age and area after its reappearance at Lyri. So the core assumption of our study is the possible distinction of two reference periods separated by the so-called missing years, and two geographical areas characterized by distinct human lineages and plant species, which we can trace by DNA analysis. A few words on the samples we have used for DNA testing. Those samples were provided by Giulio Fanti in 2010, who in turn received them personally in 2006 from Giovanni Riggi di Numana, the one who was authorized to collect samples of the shroud in 1978 and 88. Very small specimens containing microscopic particles were vacuumed from the backside of the shroud and from the main parts of the body image, and also on the corner used for radiocarbon dated. Those specimens were collected from the official filters under sterile conditions, were immobilized on adhesive tapes and attached to microscope slides. Here you have a schematic representation of the specimens from the lateral edge and internal parts, filters corresponding to the hands, face, glutei, and feet. And we used those samples, microscopic samples, for DNA isolation. Just to give you an idea, here are some pictures taken under or using an optical microscopy, showing you some pollen grains, spores, plant-like particles, cell debris, and also blood-like clots. The same specimens were also analyzed by scanning electron microscopy. And here you can have details of pollen grains, spores, plant fibers, and different types of cell debris. We used all those very small microscopic particles, contaminations, for genomic DNA isolation. So first of all, we set up a protocol, a robust protocol to isolate and sequence DNA molecules from those microscopic biological contaminants of the shroud. Then the three main goals were detection of plant DNA, chloroplast DNA, and also animal and eventually human DNA, mitochondrial type DNA. Second, determination of the number of taxonomic entities, plant species, animal species, other organisms, including possibly haplotypes and haplogroups for human subjects. Last but not least, the main goal was the definition of the geographic origin and the ethnic origin of our samples, and eventually perform a correlation of our molecular data with the historical information available for plant species and human lineages. I don't want to give too many details on the methodologies we used, but let me tell you that we adopted chloroplast DNA barcoding and mitochondrial DNA haplotyping, which are very powerful, robust, and reliable methodologies for uncovering plant taxonomic entities and human ethnic groups. Usually DNA markers belong to the extranuclear genomes, like the chloroplast and the mitochondrial genomes. We selected very standard, universal DNA markers for DNA testing. For plant chloroplast DNA, we used 3 very conventional markers: the Rubisco gene, the intergenic spacer, tRNA, PSBA, and the intron of the gene encoding for the transfer RNA of leucine. For human mitochondrial DNA analysis, we adopted the control region, the so-called D-loop, containing the hypervariable region 1 and 2, and also part of the genes encoding for dehydrogenase subunit 5 and cytochrome oxidase subunit 1. So the strategy was very simple. After isolation of genomic DNA from pollen grains, natural fibers, and cell debris, the genetic identification of plant species was carried out by DNA barcoding, chloroplast DNA barcoding, by exploiting those 3 barcodes I was mentioning. Here you find between brackets. Similarly, for human DNA specimens, we isolated genomic DNA after purification from organic particles, including thin blood crusts or clots. We were able to sequence DNA markers by using an approach called mitochondrial DNA haplotyping for the control region and the 2 genes we selected for sequencing. So, to make a long story very short, this is the project pipeline. Target regions were PCR amplified, and the Amplicons of the plant chloroplast DNA barcodes and human mitochondrial DNA markers were used for sequencing using conventional methods like Sanger method or next-generation sequencing method like the 454 method. After sequence editing and database querying, a number of bioinformatic tools were used for the genetic identification of plant and animal species, and also for their quantification. This is an important part. Usually, ancient DNA fragments are very short, shorter than 200 base pairs. But the size of those fragments, of the ancient DNA fragments, very often ranges between 100 and 500 base pairs, according to a number of parameters, including time, pH, temperature, moisture. So, longer DNA sequences are usually associated to modern, recent contaminations. Even if we cannot rule out that Amplicons overlapping that size range could be related to old contaminations and derived from ancient DNA templates. In our study, in addition to long DNA sequences, which are usually associated to modern contaminants, we detected many short, very short DNA sequences, shorter than 200 base pairs, which can likely be due to ancient contaminations. In particular, for plant species, chloroplast DNA sequences were ranging between 229 and 622 base pairs. For human sources, mitochondrial DNA sequences were ranging between 419 and 576 base pairs. But fragments shorter than 150 base pairs were very often found. So we got DNA sequences related to ancient, but also to modern contaminations. Here, the first part of the results in terms of species distribution across filters of the Shroud. We were able to identify at least 19 taxonomically different plant species, at least 21 genetically different human subjects. In addition to a small bird called southern great shrike, which is Lanius meridionalis, and also to a marine worm, which is Cerebratulus longiceps. In addition, we found many bacterial microorganisms, many fungal organisms, including Malassezia globosa, which is the fungal agent responsible for dandruff in humans. I'd like to stress that the lateral edge, the filter in the corner of the shroud, was the richest in plant DNA, and that human DNA was found abundant across all filters, exception made for the filter E, the hands. And I know that Giulio Fanti has an explanation for this. This table summarizes all species we found, at least 19 plant taxa. Maybe you cannot read it, but I'd like to emphasize some important points. First of all, again, DNA sequences were ranging between 229 short sequences and 622 long sequences. And E values were always very low, and expectations for genetic identity very high, 99 or 100%. So molecular data for the genetic identification of plant species are very robust and reliable. Most importantly, the same species was often identified in different filters of the shroud, a sort of biological replicate, like, for instance, Norway spruce or black locust. The same species was also identified by sequencing different genes or different barcodes, a technical replicate, like, for instance, clovers and hornbeams. About missing parallels. To the best of my knowledge, our molecular data did not match with any of the plant species identified on the Shroud of Turin and the Sudarium of Oviedo based on pollen grain analysis. Several of the plant species we identified are native to the Mediterranean basin, like clovers, raygrass, horsetail, plantain, and chicory. Some others have their primary center of origin in the Middle East, like wild grapes and hawthorn. Other species have their primary center of origin in Central Asia, including China, like common hop, wild forms of plums and pears, hornbeam and walnut. Other species we know to be widely present and widespread in Central and Northern Europe, like Norway spruce. Some others have been introduced from Southern and Northern America in recent centuries, like members of the Solanaceae family and black locust, which is Robinia pseudocacha, a legume plant. This is one of the figures, the original figures of the article showing you don't need this, right? Okay, I was saying that this is one of the original figures of the paper showing a schematic overview of the Vavilov centers of origin for plant species identified on the Shroud. So I'd like to stress once more that in addition to plant species native to the Mediterranean basin, we identified several taxa with a center of origin in Asia, the Middle East, or the Americas, but most likely introduced in a historical interval later than the medieval period. For instance, we know that black locust, which is Robinia pseudacacia, was introduced in Europe in the 17th century. And we also know that some wild forms of the Drupaceae and Pomaceae tribes are plants introduced in Europe from China in the 13th century. So, historical interval later than the medieval period. Let me also add that the southern gray shrike, the small bird I was talking before, has a distribution area in northern regions of Africa, from the Iberian Peninsula to the Arabian Peninsula. The most abundant plant DNA we found in the Shroud belongs to Pichia abies, which is Norway spruce. A number of diagnostic single nucleotide polymorphisms allowed us to discriminate Norway spruce from other very similar species like Serbian spruce or Siberian spruce. We know that the DNA sequencing, the sequences are quite long, longer than 500 or 600 base pairs. So this finding suggests that they are coming from recent or modern contaminations. Interestingly, the spruce tree, genetically more similar to the ones we found in the Shroud, among the contaminants of the Shroud, is located in the Swiss Alps. Here is the voucher of that plant, very close to Chambéry and Turin. So again, this finding suggests a very recent contamination. What about human mitochondrial DNA haplotypes? We identified multiple subjects. A total of 38 different haplotypes, which allowed us to predict 14 main haplogroup types and at least 21 sub-haplogroups. This is another original figure of the paper showing a schematic overview of the geographical distribution of human haplogroups and sub-haplogroups. Our data suggests that DNA sequences can cluster into a number of Eurasian ethnic groups, including some known to be typical in Western Europe, like U5b, or the Iberian Peninsula, like H1 and H3, Northeastern Africa, L3c, or the Caucasus and the Middle East, like H2a and H13, the Near East, H33, the Arabian Peninsula, R0a, and unexpectedly, several human haplogroups of the Indian subcontinent, like M39, 56, R8, and 7. So all filters of the Shroud, the lateral edge, the internal parts of the Shroud, revealed large amount of DNA related to Indian tribes, especially M56 and R8. Regarding the Middle East area, the most abundant DNA types, H2A, H13, and 33, are mainly found in the Caucasus and Near East regions. Here, in the Near East, from Egypt to Iran, Iraq, the northern part of the Arabian Peninsula up to Anatolia, especially H33, which is a very rare haplogroup belonging to the Druze community. These are original statistics, not yet published, regarding haplogroup frequencies and geographical areas. About total DNA distribution, I'd like to show you that DNA from Western European populations is less than 6% of the total DNA we recovered on the Shroud. Near East populations, the total DNA is almost 56%. The vast majority of DNA, mitochondrial DNA, belongs to Middle Eastern populations, and 39% from Indian tribes. So, 3 main points. The Shroud, if the Shroud is a medieval fake forged somewhere in the 14th century, it is difficult to explain why human mitochondrial DNA associated to Western European population is so scanty, less than 6% of the total DNA. What about the Indian haplogroups? 39% of the haplogroups belongs to human tribes of the Indian subcontinent. We should explain this finding. The most abundant human haplogroups are attributable to ethnic groups of the Near and Middle East area, where the presence of the Shroud was hypothesized until 1204. And these 3 I was mentioning before are the 3 most abundant haplogroups, H2A, H13, and 33. I will come on these 3 haplogroups later. So, coming to the conclusions. This is a very important, relevant evidence. The most abundant human mitogenome lineages totally overlap the Near East geographic areas where the presence of the Shroud was historically documented or hypothesized up to the sack of Constantinople. Western European lineages are represented by less than 6% of the total genomic DNA found in the Shroud, even if the Shroud has been permanently maintained in France and Italy in the last 7 centuries. H33 here is a very rare haplogroup, mainly found among the Druze community. Druze populations nowadays present in Israel, Jordan, Lebanon, Syria, here. And 39% of the haplogroups are from the Indian subcontinent. So, a possible explanation, and we formulate a new hypothesis on this, is that an Indian origin of the linen and a Jewish manufacture of the cloth. In our paper 2 years ago, we wrote that the detection of mitochondrial haplogroups that are typical from India is somehow unexpected. One obvious possibility is that during the course of centuries, individuals of Indian ancestry came into contact with the Shroud. But one alternative possibility is that the linen yarn was imported from India, as supported perhaps by the original name Sindon, which appears to derive from Sindhya or Sindhian, a yarn or fabric coming from India. That was our comment and conclusion this. A few months later of our publication, I was contacted by Ada Grossi, who wrote me after having read my paper in 2012 during some studies on the textile characteristics of the Shroud in relation to the ancient Jewish funerary uses. I made the assumption that while it is almost certain that the weaving of the Shroud occurred in a Jewish environmental, the linen yarn was likely imported. More specifically, I think it is an Indian production linen yarn, which in rabbinic sources is named Hindain, a word describing yarns, not fabrics. So different approaches, completely different strategies, studies, supporting a similar hypothesis. On my concluding remarks, I'd like to comment on the possible haplogroups of the domain of the Shroud. H13 is a relatively common human lineage shared by population around the Caucasus, mostly found in the Near Eastern regions, up to 3%. Our research has found such DNA type mainly in the lateral edge of the shroud, while it is scanty in the internal parts of the body image. The second most abundant haplogroup is H33, which is a very rare human lineage. As I was mentioning, it is found among the Druze community. Relatively recent paper, but an important paper, found that Druze populations share a common genetic ancestry with Jews and Cypriots and have historically exchanged their Levantine genes with Near Eastern populations, including Palestinians and Syrians. Those data were published on Nature, a very important journal. The last abundant haplogroup is H2A, which is including the reference sequence for mitochondrial DNA in human, and it is found all over Western Eurasian regions, with the highest frequencies in the Caucasus, up to 5%. Interestingly, this haplogroup is the most common DNA in the internal parts of the body image of the shroud. In our paper, the conclusions were centered on two possible scenarios for the Shroud of Turin: a medieval forgery or an authentic burial cloth 2,000 years old. In the first case, most DNA contamination should derive from ethnic groups from the Near East, the Caucasus, Anatolia, eventually India, which came into contact with the shroud in Western Europe, France, and Italy. In the second case, we can say that different folks or devotees came into contact with the shroud during the long journey from Jerusalem to Turin, across their own lands and cities, populations, local populations, and different historical ages. Here, please give a look to this comparison. Here we have the relative amounts of mitochondrial DNA haplogroups, less than 6% in Western Europe, 56% in the Near East, 39%, almost 39% in India. Here, the distribution of mitochondrial DNA haplogroups. So, if it is true that such genetic diversity does not exclude a medieval origin for the shroud in Western Europe, it is also true that our molecular results are fully compatible, totally compatible with the historic path followed by the shroud during the long journey from Jerusalem to Turin. I'd like to thank, first of all, Giulio Fanti to have provided the shroud samples, Giulio Galla, who is the senior collaborator in my research group, the one who produced the vast majority of DNA sequences, and also the research group of Professor Antonio Torroni from the University of Pavia for the analysis of human mitochondrial DNA sequences. This research was supported by the University of Padua. Thank you for your attention.